All right. Okay. So, today we are going to continue our journey through the cell with cytoskeleton, which we usually in the past have just described as a bunch of like little scaffolding things. But we are going to kind of dig a little bit deeper. So we're going to talk about some of the cytoskeletal elements today, a few of them tomorrow, and then we will do an activity Friday and Monday. So again, will not all just be me talking, but the goal for today is for you to be able to describe the functions of the cytoskeleton and use elements of their structures as evidence to support your initial description. So tell me why or how the cytoskeleton works and why its shape allows it to do that work. So to start us off, there are two big functions of the cytoskeleton. That is mechanical support, so actually physically supporting the cell. This is really important, not recall important, really important for animal cells who don't have cell walls to do all of their support for them. There we go. This is really important for animal cells without cell walls because in a plant cell and some bacteria cells, those cell walls do a lot of the work of holding that cell up. But animal cells are kind of blobby, and they get most of their support from their cytoskeleton, like we get most of our support from our skeleton. And they're also super important for movement. That is, movement in and outside of the cell. So, that is a cell moving around physically, like getting from point A to point B, and moving cell parts inside of the cell around, like our vesicles moving through our cells to get from one place to another. The cytoskeleton has a really important role in that. This typically requires interaction between cytoskeleton and motor proteins. So there are motor proteins that are really involved in these processes. We're going to talk about one of them in particular. And all of those kind of work together to get things like cell movement in and out of the cell. We're going to talk about some really cool examples today. I'm kind of excited about it. So, maintaining cell shape and moving cells around. That is what our main functions of the cytoskeleton are. Two big roles. And that word cell motility, you hear that word motility, it's not just the cell like moving around physically, it is also how well the components of the cell can move inside of it. There are three big types, or three major components of our cytoskeleton. There are microtubules, microfilaments, and intermediate filaments. Microtubules, microfilaments, and intermediate filaments. And you can see this kind of diagram right here has our microtubules in blue, our intermediate filaments in a yellowish orange, and our microfilaments in green. So these are our three big components. We're going to focus on the major one today, and we'll talk about the two smaller ones tomorrow. So these are our big components of our cytoskeleton. So just these three. Today, we're talking about microtubules. Microtubules are the one of the more common, one of the more important, one of the more studied components of the cytoskeleton. Their shape is actually really cool. They are hollow rods constructed of these globular proteins called tubulins. So microtubules are made up of tubulins, and they are dimers. A dimer means that there are two subunits, an alpha and a beta subunit. Now, sometimes you might see the alpha and beta written out like this weird little A thing, and this B with a really long tail. Alpha and beta, Greek symbols. I wrote them out because I didn't find the characters for them. Or I just wanted you to have the actual words involved. Those are the, so you can see them right here, actually. So we've got our alpha tubulin and our beta tubulin. And they work together in these little pairs, and these little pairs will wind up on one another and build these really long tubes. And in order to grow or shrink the tube, all you have to do is add or subtract these tubulins. So if we wanted to grow out our microtubule, we just tack more tubulin proteins or tubulins on there. Yeah, proteins. And if we were to break them down, we can just take those tubulins off the ends. So there are, this is our cross section. So if we were to look at it down the barrel, it's just like a little ring. So we're just making a big long hollow tube of these proteins. So a cell that has a lot of movement involved with it, who has a lot of cytoskeletons, who has a lot of microtubules, might also have a lot of endoplasmic reticulum and ribosomes, because those ribosomes have to build the proteins that build up our microtubules. So they kind of play in on one another. But these are our big microtubule shapes. All right. Their functions, specifically microtubules. So the first functions we talked about were like the generalized function of the entire cytoskeleton. These are our microtubule specific functions. So their main goal, shape and support the cell, right? Our cells need structural support, but they also serve as tracks where organelles with motor proteins can move. So there are these microtubule tracks that lead from the ER to the Golgi, because our ER doesn't target our vesicles, right? Our ER just sends them to the Golgi, and then the Golgi will tag and ship them to where they need to go. So there's kind of like a straight shot from the ER to the Golgi made up of these microtubules. They're also really important in cell division. So cell division is probably where you've heard the most about microtubules and cytoskeletons, because they are, whenever we're drawing mitosis, the little finger lines that like move everything across the cell. So these microtubules have really, really critical functions. If the cell could not make microtubules, the cell could no longer divide, because it doesn't have the tracks on which our cell division is going to happen. If they didn't have microtubules, the protein manufacturing and distribution would be completely messed up, because we don't have a path for our vesicles to slide across. So these are kind of effervescent in our cells. They're really, really important to a lot of cell functions that go on. So we're going to talk about specific types or structures that are built out of microtubules. The first of which are our centrosomes and centrioles. Centrosomes and centrioles. A centrosome is a pair of centrioles in a region often located near the nucleus. That is the centrosome. It's a region near the nucleus. So here's our nucleus, here's our centrosome in our little cell. And it has two centrioles. A centriole is 9 sets of triplet microtubules in a ring. So if we were to draw that out, we'd have a, gracious, of course. Pull the lid off that for me. Thank you. So if we wanted a set of 9 of these microtubules, and we wanted to draw a centriole, it is 9 sets of triplets. So each one of these has 3 coming off of it. Now theirs are a little bit more like crisscrossed, but essentially this is our centriole. 2 of these makes up our centrosome. So we've got microtubules making up the centriole, making up the centrosome. Lots of words that sound very, very similar. Back to back to back to back to back. Microtubules organize themselves. So the pictures on this last slide, pictures on these. This tube right here is each one of these little circles. That makes sense? All right. So our functions of our centrioles. In animal cells, our centrosome might be where our microtubules grow. We don't know 100% for sure. They also may, help support. The word support is in here. There we go. They help support our microtubules in animal cells. But the reason we don't know this for sure is because some eukaryotes don't have a centrosome. And they organize their centrioles in some other way. So we think that this is our big role of our centrosome. We know that they can help in cell division. And usually when we draw out mitosis, we draw out our little centrosomes. But that is not the case in every single cell. Like they can do the processes that cells with centrosomes do without centrosomes. So we're not 100% sure, but we're pretty sure that it has something to do with microtubule organization and microtubule manufacturing. So these are our centrosome centriole major functions. Remember, we're supporting our cell, we're helping cell division, all that fun stuff. Now I think we're getting to the fun stuff. Cilia and flagella. Oh, did I move too fast? No. Okay. Cilia and flagella are like 2 of my absolute favorite organelles. They have very, very similar structures, so we're going to talk about them as a group, and then I'll do some differentiation. What they are is a group of microtubules, so a bunch of microtubules that are wrapped in an extension of the plasma membrane. So the plasma membrane is still going to enclose them. They usually jut out of the cell. They're anchored to the cell by something called a basal body, which is kind of shaped like a centriole. I got so excited about the fun fact that I missed this. Shaped like a centriole. What's super fun about this, and which I think is beautiful, is that so sperm cells have little tails. Those little tails are the flagella. The basal body of that flagella, so the thing that is connecting the little tail onto a sperm cell, becomes the centriole in a fertilized egg and leads that egg through the first mitotic division, which is so cool because we are reusing something that was originally just for cell transport into like the first division of what could possibly become a new organism, which is super cool. So we've got our extensions of our cell plasma membrane that are sheathing up a group of microtubules, and they're anchored to the cell by something called a basal body. That basal body is shaped like a centriole, and in some cases will eventually become a centriole, which I think is super, super cool. So these are our cilia and flagella. They have they're kind of very, very, very similar. But like I said, we'll do some key differences, typically with like size and stuff. So you don't have to write down the fun fact if you don't want to, but I think it's really, really cool. So I've got a picture on the next slide. We've got our microtubule wrapped in our plasma membrane, and we've got this like thicker centriole shaped thing holding it in place, just like this. So this is going to be our cilia or flagella. It'll come out to a point. They've just kind of cross-sected it, bisected it. And this down here is our basal body. So this would be like the rest of the cell. We've got our basal body down here connecting all of the microtubules that are in our cilia and flagella. So this is what we're looking at. Now, what do they do? Lots of really cool stuff. So many like single cell eukaryotes are propelled through fluid by the by cilia and flagella. So like our kind of idea of a sperm cell, they like swim. But a lot of unicellular eukaryotes also just move through fluid. But sometimes cells that are held in place also have cilia and flagella. So cells that aren't meant to move have these cilia and flagella, and their goal is to move the fluid around them. So, for example, there are cells in the lining of our tracheas that have cilia and flagella, and they move in a certain direction to help us get mucus out of where it's not supposed to be, in our trachea, right? Trachea is really important. We don't want it to get all clogged up because then we can't breathe. So the cells that line the trachea have these, I think it's cilia, that will move in a certain way to help get the mucus out of our tracheas. During ovulation, there are cells in the oviducts and fallopian tubes that have cilia and flagella that actually are in charge of moving eggs to the uterus, which is kind of not something you really think about. We just like think about them moving, but it is flagella that are pushing our eggs towards the uterus, which is kind of cool. So very, very multi-purpose little tools, right? They can do anything from move fluid down in one direction, like little conveyor belts, to like actually swimming and propelling cells through fluid. Super cool. All right. So we're going to compare and contrast a little bit. Instead of a Venn diagram, I just have like two main points. Flagella will usually only, like a cell with flagella will usually only have one or two, like because they're typically a lot bigger than cilia. Cells with cilia have a lot of cilia, a lot of those little fingers. And the way they move is also really different. Flagella will move in like an undulating propeller-esque stuff, like a fish. So we've got like the little swimmy stuff moving in a circle. But the cilia will do alternating power and recovery strokes. So think about the mechanism of rowing a boat, right? You've got one really, really hard stroke one direction, and then a nice, easy one in the back, and like back and forth, and back and forth, and back and forth. So the way they move is really different too. They both can do things like move fluid and move through fluid, but how they do it is a little bit differently. Because if you've got a lot of cilia, think of it like a lot of rowers on one boat, you can get a lot of stuff done just in like smaller movements, where we've got our flagella, singular flagellum, that will kind of move like a fish. So the movements are super different too. Again, we've got just a diagram of our movement. Our cilia or cilium are going to move back and forth with power and recovery strokes, and our flagellums are going to kind of swim like that. Now, cilia have one more function than flagella do. Cilia have an extra like bonus function, and that is in signal receiving, which I think is super cool. They almost act as antennas, and these antennas don't move. They don't have the ability to do the power recovery stroke thing because they're meant to receive signals. Usually, when we do have these cilia that are meant to receive signals, there's only one of them in a cell, which is uncommon for cilia. Usually, flagella is the one with just one. And we call this the primary cilia. These are super important to brain function, right? Because our brains are just a lot of signals being sent and received. And they're also super important to embryonic development because we've got a lot of signals being sent whenever embryos are developing because we get a lot of stuff going on. So sometimes cilia can act as antennas, and when they are acting as antennas, they are non-motile. They do not move. So like I said, all of these structures, our centrosomes, our centrioles, our flagellum, our cilium, are all made of microtubules. And that means our microtubules are involved in a lot of different functions, from moving mucus out of our tracheas, to sending and receiving signals during development, to propelling cells through water. Cilium and flagellum have a lot of special functions. Now, one or two more left. We've talked about like how physically they move. We haven't really talked about like mechanically how they move. So I think that's going to be next. I'll give you a second. So our cilia and flagella are going to bend, and my picture is, of course, going to be taking up a lot of my space because why wouldn't it? What's going to happen is when we look at this cross section that we talked about right through here, each of these microtubules have these special motor proteins called dyneins. These dyneins have these little feet that will walk up and down these microtubules. So if we have two microtubules next to one another, actually, I'll wait till you're ready for the every time one of these little feet take a step, they're going to use a molecule of ATP for energy. So anytime one of these cilia or flagella move, we are using a lot of energy, a lot of ATP, in order to get that cell going, in order to start this movement. So every little step that these dianins take will move these microtubules. So right here, we've got our microtubules and we've got our dianids attached to them. What happens is one of these is going to lock up the other, and it's going to pull it down until eventually our microtubules bend. They'll go back and forth and back and forth and back and forth, and that's how we get our motion. I'll come back to this slide in a second, but I want to show you how these proteins work. They have little feet that are going to lock up this microtubule. And these up here are going to be attached to a different microtubule. And what happens is as this moves up, it's going to bend our tubules over. And another one right here, this one is doing something else that's not part of microtubules, but it's another rendition of how our dianins, each step, just move bit by bit by bit forward, which I think is really, really cool. Does everybody get this? Okay. Let's see. That is all of the new content I have for you today. Let me see if there's time for questions. Okay. So I want you to start thinking about these questions, and we will finish them up tomorrow. So I will actually just go ahead and post them on Canvas really quick, and we'll do it like an exit ticket, just because time is wonky. So just give me 2 seconds. I will freeze these up here so you can read them.